So our fourth objective, to understand regulation and control of cardiac output, really talking about how we can apply our knowledge of the autonomic nervous system and talk about how we regulate this stuff. How do we increase heart rate? How do we decrease heart rate? How do we send more blood to our vasculature, more blood to our organs? How do we send less blood? And so we're going to talk about how we regulate the cardiovascular output and how the autonomic nervous system, parasympathetic and sympathetic, have a role to play there. So let's talk about what cardiac output is. It's the amount of blood that's pumped out by each ventricle per minute. So it's a rate, essentially. So we look at this equation here for cardiac output. It's our stroke volume. Remember, stroke volume is how much blood is ejected, how much blood leaves the ventricle, times our heart rate, which is how fast the heart is beating, right? And so when we think about what can influence our cardiac output, we then have to think about what can influence our stroke volume and what can influence our heart rate as separate metrics, right? So we'll talk about heart rate factors, what can influence heart rate, and then we'll talk about what can influence stroke volume. Now, our average cardiac output at rest is about 5 liters, which is pretty amazing. Our total blood volume is about 5.5 liters. And so what that means is that in one single minute at rest, we're pumping our entire blood volume out of each ventricle. Really, really amazing how hard the heart works, how consistently the heart does this. Okay. So let's talk about some of the factors here. So we've got heart rate, how fast the heart is beating, stroke volume, how much blood is being ejected. And so we're going to consider what can influence those two separate factors as a product of what can influence the cardiac output overall. So it's going to be regulated by both intrinsic and extrinsic factors. And we kind of talked about this idea before. Anything extrinsic is outward influences, so neurons, hormones, neurotransmitters. That's what we call extrinsic regulation. And then intrinsic regulation is how the heart regulates its own rate and rhythm, or its own contractility. We'll talk about the factors where the heart can determine stroke volume particularly. So the first thing we want to talk about is the autonomic inputs to the heart. So we've got sympathetic inputs, we've got parasympathetic inputs. When we talk about sympathetic, and again, this should be sort of a review for us at this point, right? We know that we've got fibers, preganglionic coming from the spinal cord, synapsing onto our sympathetic chain, and then postganglionic fibers making their way out to the effector tissue, which is specifically the heart here. The sympathetic division of the autonomic nervous system influences 3 factors of the heart. It influences the weight of the SA node, the weight of the AV node, and then the myocardial contractile cells specifically. So how hard those contractile cells are producing force. The parasympathetic system, on the other hand, only influences the SA node and the AV node. There is no parasympathetic influence or input onto the contractile cells themselves. Okay. Let's talk about the heart rate first. So how do we influence heart rate? What is heart rate? It's going to be determined by how fast the SA node is firing, right? Remember, we said that the SA node is that cluster of pacemaker cells. It's setting the entire tone in terms of their heart rate and heart rhythm. And so if the SA node has a firing rate, we talked about it being about 100, not in our physiology, with no extrinsic control. If we took an SA nodal cell, we put it into a petri dish, it would have action potentials, 100 of those each minute. Because it's under the influence of our physiology, and because at rest there's slightly more parasympathetic influence, this brings the SA nodal firing rate down to about 70, 75 per minute. And that's where we get our resting heart rate from. If we're excited, if we're, fighting or fleeing or freezing, if we're under that sympathetic influence, the sympathetic is going to slightly take over, and that's what's going to get our heart rate up to possibly 100 beats per minute. Let's talk about how that happens. So we see increased sympathetic activity, and we want to think about the backstory here, right? Think about the connection. We know that the sympathetic activity is coming as a means of instruction coming down from those integrating centers in the brain. Let's say you stand up really quickly, your blood pressure drops, there's an afferent signal sent to the brain that we need to increase blood pressure. We send those signals to the medulla oblongata. The medulla oblongata makes the decision to send on efferent signals the instruction to increase the heart rate. And so what happens is The receptors that are on the heart, we know that the specific receptors found on the heart are always beta-1 receptors. They're going to receive more of the epinephrine that's being released from those postganglionic neurons at the neuroeffector junction. And then that is going to increase the opening states of our sodium channels, our calcium channels. And we know that the opening of these two types of channels lead us to EPSPs, right? Excitatory potentials on the membrane. And so that's going to increase how fast we spontaneously depolarize this cell, the cells here. And that's going to increase our heart rate. And I want you to bear something in mind here. We're not depolarizing these cells. We're not depolarizing these SA nodal cells. Remember, these cells can spontaneously depolarize on their own. What we're doing is we're making it easier for them to generate the next depolarization because we're keeping these channels open. Remember that the membrane permeability and the membrane potential really is a reflection of what channels are open. And if we can increase the state of our sodium channels and calcium channels being open more for longer, we're then going to make it easier for these cells to spontaneously depolarize, and they're going to do so more easily and more rapidly, and that's what increases the heart rate. Okay. So here's an illustration of that, right? We've got our norepinephrine or epinephrine crossing that junction, either from a postganglionic cell or circulating in the blood as a hormone. So we know that it can be either way. It can bind to our beta-1 receptor, again, making that connection that beta-1 receptors are the specific adrenergic receptors that are located on the heart. This is a stimulatory G protein that it's coupled to. It's going to have that downstream effect, turn on this amplifier enzyme, adenylyl cyclase. That's going to create our second messenger here, which is cyclic AMP. That's going to turn on protein kinase. And we talked about this part of this second messenger pathway already in more a general way when we looked at these receptors. And so we're gonna kind of fill in the specifics of what exactly this protein kinase does. So it basically opens up funny channels, opens up our calcium channels, and makes it more likely, more favorable, easier for the cell to spontaneously depolarize. Okay, and if you remember, the funny channels were the channels that kind of pretty much initiated that initial depolarization that allowed the pacemaker cells specifically to depolarize. Okay. Let's look at how the parasympathetic system influences the heart rate. So the parasympathetic signals are arriving to the heart via the vagus nerve, right? We know that that's one of the parasympathetic neurons. It's going to release its neurotransmitter, which is acetylcholine here. That's going to bind to this muscarinic cholinergic receptor. Again, thinking back, making the connection back to what we talked about at the neuroeffector junction. On the parasympathetic system, we've got the muscarinic cholinergic receptors only, excepting sweat glands. And so we bind that acetylcholine to these muscarinic cholinergic receptors. And what this does is it increases this opening state of potassium channels, and it closes the calcium channels. And what that does is it makes it really hard for this cell to spontaneously depolarize. It kind of keeps the cell in this hyperpolarized state. And so in order to depolarize, the cell is going to have to work even harder to overcome that hyperpolarized state that's being created by opening up these potassium channels. Remember, when potassium channels are open, that's going to allow potassium to leave and kind of keep the membrane potential more negative. Likewise, if we close calcium channels, we're going to make it hard for that initial depolarization to happen. And so these events are going to decrease the heart rate because of this neural signal that's coming in from the parasympathetic pathway. And again, illustrating that here, we've got acetylcholine being released from our postganglionic neuron. Remember, this is the neuroeffector junction specific to cardiac muscle. It's going to bind to a muscarinic cholinergic receptor. And we have this pathway here, which we're not really looking at the specifics, but we're connecting that the outcome of this pathway is to close calcium channels, these T-type calcium channels, and then open up potassium channels. And these two events make it really hard to then depolarize the cell. Okay, lots of potassium will start leaving, so it's going to keep the cell hyperpolarized. And if we can't get calcium in, we can't have that initial depolarization that we know is needed to bring about the action potential in a pacemaker cell. Okay, And so kind of making all of that connect, let's look at this graph here. We're comparing the membrane potential to time here on the X. And what we see is that the curve in gray shows the amount of action potentials that we can fire in this given time at rest. So if we have even inputs of both sympathetic and parasympathetic systems, we can fire about 2 action potentials in this time frame. If we have parasympathetic influence, we actually only fire 1 action potential in this time frame, again, because it becomes so hard to depolarize that cell because of the changes that are brought about by acetylcholine binding to that muscarinic receptor. And then on the other end of the spectrum, it becomes really easy to spontaneously depolarize this pacemaker cell if we have sympathetic influence. And so we can fire 3 action potentials in this same given time frame. So that's kind of the neural input on heart rate. Let's talk about the hormonal inputs to heart rate. So there are 2 hormones that we know have an effect on heart rate. One is epinephrine, and epinephrine is going to behave just like norepinephrine binding here to this beta-1 receptor. The only difference is this epinephrine is circulating in the blood as a hormone, and we know that the source of this epinephrine is going to be that adrenal medulla-chromaffin cell. So it's going to have the same effect as a sympathetic nervous system postganglionic neuron. It's going to increase the action potential firing rate. It's going to increase the velocity of action potential conductions in the muscle fiber as well. It's just going to make it easier to spontaneously depolarize those cells and then increase the heart rate that way. Glucagon is another hormone. We'll talk more about this in the GI system next semester, but it also can increase heart rate as well. And then if we talk about the integration of heart rate control, the activity of the sympathetic neurons on the SA nodal cells raises the heart rate. The activity of the parasympathetic neurons, again, via that muscarinic receptor, is going to lower our heart rate. And so we also have the circulating epinephrine, which is acting just like the sympathetic neurons, also raising our heart rate.